This video will continue our study of carbohydrates to meet our learning objective to describe and identify the four major biological macromolecules. After we can appreciate that monosaccharides exist as isomers in the L or D form, in the alpha or beta form, we can then study how polysaccharides are formed. Oligosaccharides are made from monosaccharides bound to one another. Specifically, it's usually the C1 of the monosaccharide covalently bound to a hydroxyl group of another. Each bond is made by condensation or dehydration reaction. It forms a carbon-oxygen-carbon linkage and is properly called a glycosidic bond. Again, the type of bond between monosaccharides is a glycosidic bond. Oligosaccharides are small groups of monosaccharides bound together by glycosidic bonds. They are sometimes simply called oligos, and may be as small as 2 sugars bound together, called a disaccharide, 3 a trisaccharide, 4 a tetrasaccharide, 5 a pentasaccharide, and so on. The term oligosaccharide generally refers to a polysaccharide made of fewer than 10 simple sugars. Glycosidic bonds are categorized as either alpha or beta, dependent upon the monosaccharide form. In this example, 2 alpha-D glucose molecules are covalently linked. The linkage is between C1 of the first glucose and C4 of the second glucose. This linkage is classified as an alpha 1,4 glycosidic bond. A repeat of the alpha 1,4 glycosidic bond is shown on the left of this image. However, glycosidic bonds can form at carbon atoms other than the number 4. Take, for example, this alpha 1,6 glycosidic bond shown to the right. Beta-glycosidic bonds are also possible when linking a beta-form monosaccharide. The nomenclature is still dependent upon which carbon atoms are bound together. In this image, a beta 1,4 glycosidic bond is formed. Glycosidic bonds can cause the resulting polysaccharide to take on a fiber, have branch points, or form a helical pattern. These various shapes are shown in glycogen, which is an important energy storage form in animals, starch, an energy storage form in plants, and cellulose, a structural polysaccharide in plant cell walls. Lastly, monosaccharides can have residues other than hydroxyl groups, such as highlighted in this N-acetylglucosamine molecule. A change in residue changes the structure and function of any molecule. In summary, glycosidic bonds are used to join monosaccharides.